Good morning or good afternoon, depending on where you are joining us today in the world. We'd like to welcome and thank you for attending this webinar session on the new FDA guidance for biocompatibility of certain devices in contact with intact skin, the naughty and nice list, and is appropriate just after Christmas. This webinar is aimed at providing medical device companies the opportunity to establish or refresh their fundamental testing knowledge and thereby achieve more efficient, accurate, and effective testing outcomes. And here to present for us today is Thor Rollins, our resident expert at Nelson Laboratories. Just a few housekeeping items I'd like to take care of. If you ever miss one of our webinars or if you want to go back and review it again, you can always find them on the Nelson Labs website on the on-demand webinars page listed under education. They usually appear there about a week after the event. Also, Nelson Labs and our sister company, Sterigenics, we host 3-day educational medical device seminar events. Now, obviously, these are on hold right now, but we will be doing those again when it is safe to do so. And you can always find out about information about these events on the News and Events page under the Seminars tab. And if you want to receive notifications on these types of events, as well as information regarding testing, sterilization, biocompatibility, packaging, and other updates, you can certainly like or follow Nelson Labs and or our sister company, Sterigenics, on Twitter, Facebook, or LinkedIn. As for this webinar, we welcome all your questions, and you can submit them. You can submit your questions at any time using the questions button that you'll see on your screen. And Thor will answer as many as he possibly can during about the last 15 minutes of our presentation today. We ask if you have a product-specific question that you contact Thor directly or a member of our sales team about it after the event. So now I'd like to introduce Thor. Thor is the Director of Toxicology and E&L Consulting. He is a certified microbiologist and specializes in the selection and conduct of in vitro and in vivo biocompatibility tests. He actively speaks on biocompatibility-related topics through Nelson Labs external seminars, webinars, and trade shows. He also participates in many industry committees and events as well. He is a participating member on all Association of Advancement of Medical Instrumentation, the AMI TC 194 and 10993 ISO committees. And he plays an active role along with the FDA and regulatory committees developing standards, discussing biocompatibility methods, and voting on changes to those standards. As one of the select group of experts in the industry, Thor's participation on committees offers him insight on industry changes and helps prepare clients for changes in testing. And now that I've introduced you to Thor, we'd like to welcome him and pass the time over to Thor.
Thanks, Mike, and thanks everyone for taking some time, probably from your home, to listen to us today. I know that there could be some webinar fatigue in the industry. I know that most people are switching to webinar-based training. Maybe someday I'll be able to see your faces once again. I do miss having an audience to kind of interact with, but this is how it's going to work for today. I know one of the things I always get asked is, will these slides be available? I'm happy to give you a copy of the slides. If you send me an email after this asking for a copy of the slides, I'd be happy to send them to you. There's nothing in here that is not, that I can't give out. But like Mike said, if you miss something or you think someone else would benefit from this, or 10 months from now you have a device that contacts the skin and you thought, what did that talk about? You can find this on our website and go back and reference it. So I wasn't really planning to really go into a webinar on this particular draft guidance document. I thought it was great. I was really happy when I heard that they were coming out with it. But I thought it was pretty, just a just good document that people would use. But I kept on getting asked, and the specific questions I'll get to about it and my thoughts on it. And so I thought, well, why not just do a webinar and a white paper? So that's what we did. And then, of course, I have children, and it was on Christmas time, and I thought, wow, the naughty and nice list makes sense. So that's what I called it. So we're going to kind of get into it a little bit. This is the agenda, and it, I'll go through it. There might be some, depending on how long it takes, there might be some time for more questions at the end of this. We'll just have to see. But the first thing we're going to kind of cover is why are we seeing these types of guidance from the FDA now? And there's more than just this, right? There's other things in the works, and there's changes that are happening at the ISO level. So what's happening to initiate this type of guidance from the FDA, and what do we expect to see in the future? And that happens to be with like new emphasis on risk assessments. I think most of us can understand in this COVID world, especially with PPE being at the forefront of a lot of people's minds, there's a huge burden on reviewers. And just like everybody, it's hard for the FDA to hire and train people. So that has a part of it. Plus, the FDA is focusing on the 3 R's of animal testing, which is replace, reduction, and refinement. So that is something, and they will say where applicable or where it's needed. So they're not going to replace with a sweep of a hand every animal test, but they are focused on where we can reduce or replace or refine these animal tests. And this falls right into that kind of approach. And that does set the stage for possible future actions going forward with animal testing. The one question that I get more than anything when this came out, in fact, I don't even know how many times, what has to do with the material list, and why are ones on there and why are some of them not? And this is the question I get a lot. Stainless steel, are you kidding me? Right? Like, why isn't there metals, titanium and cobalt and stainless steel and all these other metals that we touch every day? Why aren't they on this list? And we'll talk about the possible reasons why that is. And then afterwards, like, what do I do with this now? What do I go from here? How long do I have to wait till I can use this? What if I have a metal that touches the skin? Is that completely off the table to assess? And how do I put this type of assessment together? So that's the kind of agenda that we're going to be looking at today, and the steps we're going to take. So the first thing we're going to cover is what happened to initiate this type of guidance from the FDA. What kind of led up to it. So the big thing that we've seen, and talking with any FDA reviewer, you can kind of get this sense, which is I think most of us right now in this post-COVID world, which is there's been a lot of growth in the medical device industry. There's been a lot of need for medical devices. And there's been some new type of customers in the medical device field recently that may not have the background in medical device assessments. So there's been a high number of these types of devices influxed into the medical device world. And with this comes some lackluster justifications for these low-risk devices. I've seen some, they'll go from the background that it's like, oh, this is polypropylene, so I don't have to assess it because it's polypropylene, and that's pretty much the state of their assessment. There's no follow-up on any clinical kind of evaluations afterwards, or there's no discussion about those materials or the use really, or even the processing. We'll get all into that. And the way that ISO 10993 is now set up, and we'll cover this in a minute, it puts more of an emphasis on risk assessment, which is the absolutely correct way we should go about evaluating medical devices. It should not be just a checkbox mentality, but that risk assessment is a little bit more labor intensive when it comes to a regulatory standpoint, because the checkbox was easier because you just made sure they did the testing that the ISO says to do. But now with the risk-based approach, it does require scientific evaluation. Did they address everything in their risk? Is their plan to mitigate that risk sufficient? So it puts a little bit more demand on expert review in the FDA, which is a bottleneck. So if we can get low-risk devices off that bottleneck, it helps with the higher-risk devices. And we're seeing kind of a different side of things which overlaps. We are seeing a pressure on the FDA to reduce demand for animal tests. You have the pharmaceutical industry, you have food, you have Europe and even other parts of the world that are starting to move away from animal tests and starting to prove that animal testing may not be as predictive as we thought it was, and that we have better science that does not involve animals. And so the FDA, specifically CDRH, is starting to feel a little bit of pressure on evaluating our current stance. And that's just not the CDRH. I mean, ISO 10993, we talk about that a lot, about these animal tests that we've been using for decades. Are they still the most state-of-the-art science? Are they the best way to evaluate safety, or are we just staying in our rut? And if you have not read the guidance document yet, the draft guidance document, and you want to have a great weekend this weekend, sitting back with a cup of coffee or something, there's the link for the FDA guidance document for your reading pleasure. Okay, so to kind of expand a little bit on the concepts we just talked about, one of the big things that's happened recently to impact the workload of FDA reviewers is this 10993-1 risk-based approach that has been put into the new ISO document. Like I mentioned before, I've been doing biocompatibility now for 19 years, which makes me feel really old. But when I first started this biocompatibility approach, it was a checkbox mentality. People would just go to the standard. Actually, technically, when I first started, people weren't testing very much, to be completely honest. They were going and saying, oh, this is stainless steel, we don't need to test. It wasn't until probably 10 years ago or so that we started having testing more of a checkbox approach. And so, like I said, that's easy to have anybody. I even taught my daughter how to do the checkbox approach with the chart. So it was easy for anybody just to see if the correct testing was done per the standard. That's what I heard all the time. Oh, is the correct testing done per the standard? But that's not really the way to assess safety. Like these materials are the best example. Like these plastics that are touching the skin, we touch these plastics every day. And for the most part, we don't have a whole lot of reaction to them. So if that's the correct way to go, is a risk assessment and then a plan to mitigate any risks you find. But that does take more burden to the reviewer. And the introduction of chemical characterization and tox assessments, not necessarily for the skin contacting devices, but for the other devices that have higher risks, once again, put more burden on the reviewers. They're more scientifically demanding. It takes experts with background, and then that takes time for them to review. So that puts them in each review of these chemistry assessments and tox assessments take more time than they used to with just the animal tests. So now they have higher workloads on them per submission. So there's a way to try to, they have to reduce that workload with some of the low risk devices. So most of you will probably know, this is the chart out of ISO 10993-1. This is what I'm talking about, which is the risk-based approach based off of gathering physical and or chemical information. I sometimes sound like a broken record on this one, but I just feel like it's important to talk about, which is there's a reason why we put in physical and or chemical information, not just chemical information. It's because for these devices is a good example. We're not saying that you have to test chemistry for every single device. And sometimes, especially notified bodies or others, may interpret the standard saying that, oh, we're requiring chemistry for everything. But no, the concept is that you gather your physical and your chemical information that you feel is sufficient for your device. And then based off of that information, you evaluate these other endpoints. Are they needed? It's not just do chemistry all the time. But because of this type of approach, like I said, that now we're starting to see the risk-based approach for all these devices. And really this draft document we're talking about today is the FDA spelling out, giving you the approach for these intact skin devices up here. And by the way, this goes for all, this draft guidance is for all these contact types, this short, prolonged, and permanent. They're basically giving you what they feel like is the right approach to justify these types of endpoints. So for me, and for those of you that may have attended a couple of my seminars, webinars before, I've used this example, but I think it's the best example to kind of hit home what we're talking about here, which is about mitigating risk based on how much effort to put in. And for me, this goes back to a personal experience, which is my backpacking experience. So I'm not originally, Nelson Labs in Salt Lake City, Utah. For those of you that don't know Utah, it's a wonderful place for outdoor activities, skiing and mountain biking and hiking. I moved here 20 years ago from North Carolina. So North Carolina is great, a beautiful state, but not the same type of outdoor activities as we have here in the mountains. So when I moved here, friends and coworkers told me that we're talking about how awesome backpacking in the mountains were, and they really started making it sound like the best thing in the world. And so I decided I need to learn how to backpack now that I live in Utah. So I went to the local outdoor store, and I'd never been backpacking before. I didn't know exactly what I needed for these hikes. So I kind of went overboard. I ended up buying everything you could think of for a backpacking trip. Bear traps and spray because there's bears in these mountains. Tents, big tents because I'm a big guy. Lots of food and snacks, and I might have put a couple of cans of soda in there. But when I got done and I started loading up my backpack, this is what I felt like. It was heavy. It was really, really heavy. And then when I went up hiking in the mountains, it was miserable. After just a little while, I started thinking, why do people like this? This is not very fun. It was because I was having too much burden on me for what I was trying to do. But the more and more you went backpacking, the more you realized, depending on the length of the hike and where you're going, what you need to put in your backpack. And then it became a lot more enjoyable. In fact, it's one of the things I enjoy a lot right now living in Utah is getting up in the mountains and going backpacking. So this is kind of the symbolic of just not this approach with this risk assessment, but basically all our approaches for biocompatibility, where if you put too much in these risk assessments or not enough, you end up either going really hard and not having a very good experience with biocompatibility, or you don't put enough in your backpack and you end up not being able to get where you need to go. So this is the approach that we're needing to do, and the FDA's guidance document is basically saying, we feel like you guys haven't done a good enough job helping us justify out of these low-risk devices. So we're going to give you a guidance document to help that. So that's what this whole point is about risk, right? That even this draft document is about risk of these devices. These devices have low risks, and we need to evaluate those within the testing and the risk management process, which is a very important part of this draft if you read it closely. So you should, even when you develop this type of assessment for these devices, even if you're justifying out of all the testing, there should be some type of thought process put together where you're looking at the materials, the suppliers and the processes, and I would warn you from just saying, oh, my material's on this list, I don't have to do any testing. You still have to evaluate the material, make sure you get good information, make sure that the processing is good, that you're putting in, that you're not adding any irritants or sensitizers to that device in order to kind of follow this guidance. A good example, and I'm not going to call them out necessarily, but a very popular watch brand recently, a little while ago, had some issues with irritation, and they were using a material that's on this list. So don't think that just because this material is on this list that you're done. There still needs to be some type of assessment. Doesn't mean testing, but there should be some kind of risk assessment and mitigation of those risks in some kind of plan or evaluation to be able to talk through your thought process. So jumping right into those naughty and nice lists that we talked about. The first one we're going to talk about is nice list. And I'm actually not going to spend too much time on this because, well, it just lists the materials that are nice, right? So the good thing, and the thing that I loved about this particular list is that when I was going through this draft document when it came out, I'm like, oh, great. Look at all these materials that we test every single day. I can't tell you how many times I have tested PEEK in my 19 years, or HDPE. In fact, we use HDPE for negative controls in some of these tests of polypropylene. And so this made me super excited. The other thing that really helped me out a lot is down in the fabric section. When you start looking at things like Lycra or nylon or silk, they actually are problematic in the cytotoxicity test. So if for us, sometimes these samples would fail cytotoxicity, and then we'd have to spend a lot of time and effort justifying why that didn't matter because they're very elastic, and sometimes they have certain inclusions like latex possibly or others in these fabrics that cause some problems. So having these materials on this list, this helps us kind of maybe not having to just go through that exercise of failing a cytotox test and then justifying why that's okay, where we can just do a justification up front. Now, for me, the more interesting part of this is the naughty list. And so for this one, we're going to kind of go through each one and talk why these materials are on the naughty list. And when I mean naughty, obviously they're not bad materials, but these are just the materials that are excluded, right? So the first one is the one that gets the most attention from everybody I've talked to, which is the materials that are not listed in the nice list, right? So I think we can all understand the novel materials. That makes sense. If it's a brand new material, then there's no history. But it's the next one that we get a lot of comments, which is bulk materials. And they specifically call out titanium and stainless steel and nitinol and gold. And if you look at their reasons, they say that there are known risks or the FDA does not have adequate experience with these materials may introduce toxic risks. So that second part of that sentence is obviously with the novel materials, because we know that the FDA has lots of experience with titanium, stainless steel, nitinol, and gold, right? So it has to be that first part of it, which is there are known risks. And when I've talked to people about this, they go, how's that possible? What risks are there associated with stainless steel and titanium and nitinol? Well, I want to kind of talk and hit home here is that this, even though this particular draft guidance is for polymers and specifically calls out not to include metals, it doesn't mean that metals are automatically unsafe, right? So metals can, and we'll get to this a little bit more, but metals can still go through the exact same risk assessment. And for skin contacting metals, we can justify not doing any tests. We do it all the time. So it's not like you're forced to do testing because it's not in this nice list. What it means is you just can't use this particular draft document to talk about it. But the reason behind this, based on a lot of discussion I've had, is that there are possible allergic reactions that can happen, like nickel or other things, to some of these materials that can have contact with the skin. And they just want to make sure you're covering those potentials in your assessment. The other thing is, believe it or not, and this happens not all the time, but it has happened in my 19 years, which is not all stainless steels are created equal. In fact, about 8 or 9 years ago, we had some surgical tools made from stainless steel that we extracted for a cytotoxicity test in cell culture media. We extracted it at 37 degrees for 24 hours. We also extracted them in saline for the animal test. After that time, we came back to the container and there was nothing but brown sludge in the bottom of the container. Now, obviously, that wasn't very high quality stainless steel, and it's a surgical tool, so you can maybe justify that the risk is lower. But I would assume that if it's degrading in media, cell culture media, after 24 hours at 37 degrees Celsius, that's probably releasing a lot of ions with the use. And my assumption, and some of the discussions I've had with friends of mine, are that they've seen some risks and they know some of the risks with some of these metals. And so I think it's, now not saying that the polymers don't also have risks, but I feel like they specifically left off the metals because they're worried about some of the allergy risks that some of these metals might have. So what I would say is, one, you could always, and we'll talk about this, they've given you ways to add materials to this draft. And so I'm not saying we could just continually ask for them to add metals to this draft, because I think most of us would agree that that's a low risk to have titanium or stainless steel. But secondly, don't think you can't do it. I mean, we still do this all the time. In fact, I would that's the first thing I would look at, is if I had a skin contacting stainless steel device, is looking at justifying out of the testing. I would look at the grade of stainless steel, the history of that particular grade, my processing, and if I feel like the risk is low enough, why kill unnecessary animals, right? So that, and I'm sure there'll be questions about that in the after the presentation, but those are the thoughts when it comes to metal. By far, probably the biggest question I got when discussing this with colleagues. The next one is I think pretty self-explanatory, which is stored in or in containing fluids or creams. And this is, if you think about it, these creams or these fluids, it's been stored in this container with these creams and fluids. These chemicals have leached for the shelf life of this device into those creams and fluids. And then the likelihood of those creams and fluids to be absorbed through the skin are much higher. So if there's a sensitizer or an irritant that comes off the device, it would go into the fluid or cream, and then the chance of that cream or fluid to penetrate the top layers of the derma are much higher than the physical device. So that's why those would have to be assessed. Once again, not saying you have to test it. I hope that comes loud and clear. This can still be assessed through a risk assessment, but you need to know the materials and the cream and fluid that's stored in to understand the risk to having that absorbed into the skin and exposing potential chemicals. The next one, I'm going to skip a little bit. It's just, fabricated from in situ polymerization material. So if they're polymerized over time on the skin or in an environment where these chemicals could be present, they could introduce those types of risks. So hydrogels and other things like that, absorbent materials, those all have to go through a normal risk assessment. And then testing if you feel like the risk is not sufficiently covered in that assessment. Now, this one I think is interesting, and I've had a couple of conversations. So just so you guys know, we have used this draft guidance in a couple of our assessments for a couple of our clients. And one of them, this has come up. So it says contact the breach or compromised surface, such as abraded or shaved skin, open or healing wounds. And we've had a couple of people who in their IFU that says that the skin should be shaved before the device is applied. And if that's the case, then you can't just automatically use this draft guidance document. You still have to assess it. And obviously the reason for that is when you shave the skin, it's possible you're removing some of that top layer. They could have a little bit of compromised skin from the shaving. And that could open up a potential for chemicals to be absorbed a little bit more, such as things that are on compromised or breached skin or healing wounds. So, things like wound care and things like that obviously don't fall into this type of assessment. Doesn't mean you still can't assess it without testing. You just have to do it more of a traditional route. Reprocessed single-use devices. For those of you who may not be aware, there's a whole market out there where companies like blood pressure cuffs, for example, that are maybe single-use devices, but there are companies that will reprocess those and then have a validated reprocessing procedure, and then resell that reprocessed device. Because those are reprocessed, they go through cleaning and they go through disinfection. There could be residual surface, and also there could be chemicals left behind. So because of this, and they say in this, the FDA is unaware of the history of safe use. This has been going on for a long time. I don't think that's necessarily 100% the case, but there is potential problems with reprocessing as far as residuals go. So I think that's why the main reason why that's on the list. And then the last one is adhesives. So if you have any adhesive on there, and I think a lot of us understand why this could be the case. If you've ever used bandages before or other types of device like adhesive-based kind of materials, some people will end up getting irritation or in fact sensitization from the adhesives. And so that's something that we know about, and so the FDA is not going to allow you to do that. Okay, so kind of about the main thing we just talked about. What about metals? And I just wanted to kind of hit home and give you a reference for my thoughts. If you go back and look at this, is nickel sensitivity the reason why some of these are on here, or different qualities of bulk metals? Like if you can spell out the specific quality of your metal, then maybe it doesn't belong on this list as naughty. But since they're looking at bulk types of materials, that's why I think they left off the metals. I could argue that I've seen a lot of more variation between these polymers than I have metals, but that's some of the thoughts and discussions that I've had of why metals right now were not included on this particular list. And once again, does that mean you have to test metal components? No, you don't have to apply. You can still apply this to metals or any other different materials that you have in a device that's not on the nice list. We still go through the same concept. It's just it does not fall within this particular draft document. So it doesn't exclude you from running a risk assessment on anything. In fact, we recommend on any skin contacting device to run a risk assessment because there probably are tests that we could justify out of based on the use of the device. And that goes for even like mucosal membrane or breach or compromise. All those can be assessed. It just doesn't fall right into this draft document. So the next one is what now? Okay, so now we have this draft document. Now we have the guidance that was given to us. What's our next steps? Like, how do we use it? So when you submit this type of guidance to the FDA or using this draft guidance, once again, you have to list all your materials. So even if it doesn't have direct or indirect contact with the skin, you want to list it and you want to justify why, if it has or does not have direct or indirect contact with the skin. And you want to be able to make sure that you spell out. I use pictures. I think that's the best way to do it. But you want to make it clear what materials do, what materials don't, and why you feel like this draft is able to be used for those materials. And then this is very important. You need and this goes back to that risk assessment. You need a statement kind of confirming that that material has a long history of safe use. So this goes to where you just can't say it's in the draft, I'm good. You need to do some kind of literature search or analysis where you've been able to show that it's a history of this material. And it'd be great if it was from a very similar supplier. I would also put in there that your processing has a safe history of use, because in my personal opinion, that's more impactful for these types of devices than the materials themselves. Residual cleaners, detergents, there can be irritants, things like that. So you want to make sure that you have a validated cleaning process, or at least a review of your processing to help justify why that's not a concern. And a statement confirming that none of the exclusions the naughty list applies to your materials or your device itself. And then for your ID submission only, so only if you're doing an IDE, you need to discuss the responses you got from your device during that. So when you're reporting out, you want to make sure you can discuss that there was no sensitization results, so no erythema or edema, no irritation, things like that. The interesting thing why I'm on that is I always think it's funny when we're talking about skin contacting devices and cytotoxicity, because cytotoxicity really doesn't have a great biological response for skin contact, right? So you probably won't see cytotox on skin. You'll see irritation or sensitization, but because you have that layer of derma that's very thick and rough, those skins or cells are already dead. And so if it leaches underneath there, maybe you'll see some cells die, but you may not see it like you would with sensitization or irritation. And then what should be included in your device master record. So this is what's after you've done this type of submission, the information you put into your DMR. So you need to have a statement. This is where I would put your biological evaluation plan for this. I think this puts in perfectly. So you put in there your rationale for regarding the manufacturing, based on your justification of materials and manufacturing. And then you also have to address these different controls, right? So they're basically relying on the 21 CFRs 820s to kind of help control the processing, so that you have those are the purchasing controls of the device materials, that's in control, that you have the control of the manufacturing process and the materials coming in, and then you have any corrective and preventive action process in place. You review your complaint files and the reporting. So basically, this helps them feel confident that if there was something that slipped through with your device, maybe on a residual chemical, that we'd still see it. The concept there that if we do see it, it's still pretty low impact to the patients, but they don't want you to just move on. This is the way to help control the process and also the post approval of the medical device. Okay, so I wanted to echo kind of what Mike talked about at the beginning, which is obviously this is a very small part of biocompatibility, things we talk about. If you're interested in learning more about how to do a biological evaluation plan, right? So what we talked about to help put into this type of assessment to the FDA. If you're interested a little bit more about how sterilization could impact some of these devices, things like that, we have a lot of resources on our websites, both Sterigenics and Nelson. And also, hopefully, we'll get back to doing in-live seminars so that I can actually see people smile when I say a stupid joke, and I don't feel like I'm just talking to myself. So at this time, Mike, I know we have about 20 minutes or so left. Why don't we turn it over for some Q&A?
Great. Thanks, Thor, and thank you for your insights on this important topic. We've all got a lot more, we've all learned some great information today. And yes, we have received some questions. And if you have some questions, now would be the time to submit them if you haven't already. And we're going to try to get through as many as we possibly can during this last 20 minutes. So Thor, I'll start you off with this first one here. Do you think that this means the FDA approach to this subject is actually mirroring the EU approach?
Yeah, so I do think both. I think it's a little bit of the same. So I think the EU is moving towards the US FDA, and I think the US FDA is moving more towards the EU. So obviously MDR, which I know is a cuss word out there for a lot of people, is more similar to the FDA's approach than the MDDs are. But the FDA is moving more towards Europe. And this is not a coincidence, right? So both FDA and European representatives are participating on the ISO 10993, and we're really trying to work hard to not have so many differences between regulatory bodies. Japan does this, Korea does this, Europe does this, China does this. So we're really trying to get somewhere compromise so that it's less like that. But like a good example, so the just yesterday, the new ISO 10993-23 was published. That is taking irritation away from 10993-10 and putting it into its own standard. So it's -23 now. And in that is an in vitro alternative to the animal test. It's been approved, it's been put into the standard, and it's been published. The good kind of bar to see where this goes is now will the FDA start to accept some of these in vitro alternatives? Will this start to lead us more towards acceptance of other in vitro alternatives like sensitization? So I think it'll be interesting to see what happens the next 2 or 3 years to see how much further both EU and FDA go to be more like each other.
Great,
Mike,
thank you.
Sorry, Mike, before you put in there, I also want to mention now with Brexit, we have a whole third different thing with the MHRA, right? So MHRA is looking at their own way to enforce these types of evaluations. So there is even another regulatory body now with Brexit. So that's an interesting twist to the whole thing too. Sorry, Mike, go ahead.
No, thank you. Thank you for adding that comment. This next question is a topic that I know that you enjoy answering questions about. It says the draft guidance doesn't talk about colorant. What is your take? If we have colorant, cytotesting from supplier, can that be sufficient?
Yeah, so Mike is true. It's right. So I did a whole white paper and webinar on colorants. Go to nelsonlabs.com, right? So, but the FDA's current stance on colorants is that, if it is in a limited contacting device, that as long as the extracts don't change colors or have particulates, then you're fine. But now that we're not testing these, right? So now that we're kind of going to an assessment point of view for these types of materials, now we're not extracting them. So I have had discussions with a couple of regulatory people about this, and it just goes into your risk assessment. So the colorant may not be on the nice list, but it's not on the naughty list either, right? So what you need to do is in your assessment, you need to do research for that colorant. So you need to look at history of safe use on the medical device market. You need to also, one of the things that we can help with is you can look at the molecular weight of that colorant and kind of help justify that that's so big that it's not going to just fall out of this plastic at room temperature, right? So there are things to kind of do to help that, but you do need to, in my opinion, you do need to specifically address the colorant because it's not included specifically in this draft document. So in your BEP, you should have some type of discussion on why you think the colorant does not increase the risk enough to warrant testing. So I hope that answered that question.
Oh, thank you, Thor. Okay, here's our next question. What about the process aids used when the nice materials are molded? Do we still need to test per ISO 10993-5 and -10?
Yeah, and now -23, right? Yeah, yeah. So you don't, but you do need to spell out. So I know specifically in this document they don't go into processes, right? But anytime we've submitted this, and we've only done a couple, but anytime we've done this to the draft document, we just don't talk about the materials, we talk about the processing. And like I mentioned before, for me, the processing is actually the most concerning because I will touch polypropylene all day long, but if you start putting chemicals on that, then you have more of a concern, right? So in your risk assessment, you should still go through why that material has a safe and history of use on the market, kind of like the FDA asked you to do. You should also do the same thing with your processing. And I always recommend having some kind of validated cleaning process to show you're removing any residual manufacturing materials, because then it's easier to talk to the FDA about how this is just plastic. It's not a chemical. But yes, you should go through some kind of discussion, but it doesn't mean you have to test. So it doesn't mean you have to test per -5, per 23, per 10. It's really just the assessment, and hopefully the risk can be mitigated with that assessment.
Okay, great. Okay, on to our next question. What is your opinion that FDA does not include devices with skin contact made out of basic polymer materials already tested for biocompatibility, like USP plastic class 5 or class 6, including cytotoxicity and sensitization?
Yeah, so that's an interesting comment, and I would say that they just don't call out USB class 6, and one of the reasons why I think for those of you maybe not familiar, the USP are reevaluating the use of those endpoints for the USB class 6 and class 5. So I would say that they don't specifically include those in this list, but they also don't say you can't use it, right? So you still have to give, in this risk assessment, you still have to give a history of safe use in the industry. And I would say USB class 6 and class 5 is a great way to kind of, from their suppliers, to add to that justification. So, and like I mentioned before, just because you don't have material on this list doesn't mean that USB class 6 and class 5 can't be used to help understand the risk of that material that contacts the skin. So a good example to that is if you have something that on a mucosal membrane that you automatically don't apply to this list. But if that material has USB class 6 testing, including cytotoxicity, which isn't in USB class 6, I say that that's part of the whole assessment we do up front to understand the risk of the device, and then that could be helped to justify out of testing for that particular device. So it does play a role. It just doesn't play necessarily a role in this draft guidance specifically.
All righty, we'll keep rolling along. The next question is, per your experience, Thor, following this guideline, have you seen any conflicting or unexpected decisions from the FDA?
Yeah, so this is a limited experience. So we've done 3 of these, we've referenced with 3 devices, we've referenced this draft 3 times. One of them went really well. That only one that we've really had good discussion with the FDA. The other 2 are still being evaluated. So I don't have a whole lot of experience yet. It's relatively new. But our experience before this has been very positive. So this isn't the first time we've done this type of assessment, right? So even before this draft document, we were taking skin contacting devices and going through a very similar rationale that what the FDA is spelling out here to help justify out of animal tests. We would probably still recommend a cytotox because it's a good, sensitive, and easy test. But the animal test, we would evaluate the risk of these. And the FDA was always been very, lately especially, has been very good to work with on that front. We have a pre-sub before, which this helps us not necessarily have a pre-sub for this document, but before we'd have a pre-sub, we'd go through the rationale and why we felt like the animal tests were not needed. And we'd always have really good discussion with the FDA experts. And a lot of times we're able to come up with a good compromise on not having the animal tests. So, we still are experiencing and gathering feedback, but from what limited experience I've had, it's been really good.
Okay, great. Okay, here's our next question. Can AST address the cytotox failure if it is intact skin contact only?
Sorry, can you read that for me one more time?
Yes. Can AST, looks like it might be an acronym, address the cytotox failure if it is intact skin contact only?
So it's possible. So there's a lot of ways to address a cytotox failure. And you have lots of tools out there to do so. Lots of different chemical techniques. ICP-MS is a way if you have a failure with metals. In fact, once, I hate to keep on plugging, but we did a whole white paper on cytotoxicity of metals. It's one of the ones that we see a lot of cytotoxicity with. So there's a lot of ways and tools to justify a cytotox failure, and intact skin is definitely one of them, because when you start looking at the skin barrier, right, you start looking at the top layer like we talked about, there's a lot of ways you can evaluate if the material is going to even interact with a live cell. So those are ways to justify out of the animal or any kind of cytotoxicity for intact skin. If you have a specific problem with one of those, just give us a call. We'd love to kind of work through it on the things that we've done before.
Okay. Okay, here's our next question. For a skin contacting device used 2 hours per day every day for 5 years, should this be considered a less than 24-hour contacting device or a greater than 30 days because of cumulative time?
Yeah. So it's cumulative. So what we do in our assessments is we actually do the math, right? So we say, okay, so the device is used 2 hours a day, every day for X amount of time, and you do the math. And whatever time that comes up with is the category that the device should fall into. I've actually had this discussion recently with masks, since surgical masks are such a big deal right now. A doctor or a nurse can wear a surgical mask maybe a couple hours at a time, but they're wearing that repeatedly through the day and then pretty much their whole careers. And so when you start looking at that exposure, that 1093 is pretty clear that it's repeated use over time. So you add it up. I would assume not having the math done in my head, but that would put it up in the prolonged contact timeline. So that's what we would categorize it as.
Okay, great. Okay, here's our next question. Can you give your take on how to classify a component or device as transient contacting?
Yeah, so this is a great question. Thank you for asking it. So for those of you that don't know, the new ISO 10993 did bring out some new contact types, which is transient. And it's actually very similar concept to what this draft document is, which is there are some materials or types of contact out there that is so minute, so low frequency that we don't really have a big risk with them. And once again, we didn't want people going through all the chemistry and biocompatibility on these very low types. So such contacts that we evaluate for transient are things that could be touched by the practitioner, the nurse or the doctor, or even the user of the device, but is not really intended to always be contacted. So you're looking at things that could contact the skin. A good example is I'm right now using earbuds. The wires of the earbuds are not necessarily intended to be in contact with my skin. The end of the earbuds are, but they could swing and hit my face every once in a while. So that would be like transient contact, something that could contact the surface of my skin, but not dwell there. It doesn't always happen. It just happens to be touched every once in a while. That's how we categorize transient contact.
All right. Here's our next question. If we can determine that material composition is only fabric, can we avoid doing cytotoxicity, sensitization, and irritation testing?
Yeah, so it's pretty much what this draft document spells out, right? So if you can show that what contacts the patient, so you would want in your assessment, you would want to list all of your device materials, and then you'd want to show what could contact the patient, either directly or indirectly. And if that material falls into the nice list, then this whole draft document works, right? So then you would want to go through and do a rationale on why that material is safe to use, like it has a nice history, safe of work, or history of use. You'd also want to look at the processing that goes into it, make sure there's not any residual processing that could cause problems. And then you'd want to use your post-market surveillance to help make sure things are looking good and catching any problems. And then you'd want to put together a submission that includes everything in this draft document. But for sure, yeah, that's pretty much what this draft document was made to do.
Perfect. Okay. Next one is, does Nelson prepare bio-risk assessments as a service? Will they advise if a device is eligible for a risk-based exemption from any or all of the ISO 10993 tests?
Yeah, that's something that we have a team of toxicologists and biocomp experts and chemists, and that's pretty much what they do. So they do about, 10 to 15 BEPs a week. And we look at, it's everything we just talked about, right? What's in this document. And this document was great for us because it helped us get away from having to do so much justification for skin contacting devices. We can do now more of just what this document says. So yeah, we can definitely help there. You can send me an email afterwards or go to our website. We have where to contact us for that.
Okay. We have time for a few more questions. Here's our next one. Are there any specific biocompatible requirements for components that are not in direct contact with the end user, but in the breathing path, example, a valve on a respirator?
Yeah, there is. It's specifically called 18562. So ISO 18562 is the biocompatibility of gas path devices. And that gives you the guidance on these type of devices that are technically in indirect contact with the body. So if you think of gases, they act a lot like liquids, where they will pull chemicals from materials and especially volatile chemicals or even low weight semi-volatiles, and then that will be delivered to the patient. So 18562 is the standard that is utilized to evaluate those endpoints. And unfortunately, because it's in the lungs, in the mucosal membrane, it doesn't fall within this standard. You would have to evaluate those per 18562, which is looking at particulates, and they look at volatile chemicals coming off in the gas and doing a tox assessment on those. So if you need help looking and understanding that particular approach, let us know. We have 18562 testing in-house, so we can do that testing for you.
Okay. Here's our next question. Can you elaborate on why does this guidance exclude neonates and pregnant women?
Yeah. So first off, the neonates, that's the easier one to explain. So for neonates, they have their skin. So if you want to do some research this weekend and have some fun looking at how the skin is made up and how it changes over time, neonates and even baby skin is not the same as ours, and so it's much more permeable. It has lots more it can be a lot more absorbent potential, And so the likelihood of those chemicals leaching through the skin and interacting with the tissue below are higher with neonates than it is with us old crusty adults. So for that reason, they're excluded from this. The pregnant women, it's a similar concept, although a little bit different. So because, obviously they still have very similar skins, but bodies do change during pregnancy, and the risk of having chemicals go to an unborn child, the risk of that unborn child is much greater. And the FDA doesn't have a lot of data to help show that these chemical exposures on the skin won't impact a fetus. So even though I think we can still, once again, assess that from a risk-based approach, it's not off the table that you have to test, but it just didn't fall, the FDA didn't want to put it into this document because they want you to examine it a little closer to make sure that the fetus is not at risk.
Okay, I think we have time for one or two more. So here's the next one. Is residual testing in your cleaning validation sufficient to address reprocessed single-use devices biocompatibility?
Yeah, and that's basically what we do to assess reprocessed devices for biocompatibility. So once again, it's not included in this draft, specifically excluded, in fact, but there's a lot of times that we'll look at biocompatibility. So if you think of biocompatibility of a single-use processed device, most of the time the OEM has already done their biocompatibility for that device. So that device itself has been assessed. What we need to assess is the reprocessing of that single-use device. So does the material degrade, or is there residual processing? So doing a cleaning validation or a processing validation to show that there's no residual from the process, and to show that the materials haven't degraded that now release something they did before is all part of the assessment that we would like to do for single-use reprocessed devices. So that's something that should be thought of when you're doing your risk assessment.
Okay, and here, this will be our last question.
And Mike, just so everybody knows, so I just want to let everybody know that I have these questions, and for the questions we don't get to, I will make an effort to get back to you with the answers. So sorry, Mike, go ahead.
Yeah, no problem. Thank you, Thor. So here it is. How would you address or justify the potential effect of geometric features, such as surface finish or porosity? To what extent are they relevant for risk assessment?
Yeah, so that's a great question. I would give you a high five if I was there. So in skin contact, they're not huge. So obviously they could hold on to chemicals more if there's like crevices. And so residual chemicals could be more of an issue than a smooth device. But that really comes into play more when you start looking at like implantation effects or hemocompatibility effects. The surface characteristics are huge there, much bigger than the leachables. So that's why the FDA doesn't really spell it out in this draft, because this is for skin contacting devices. I say you still want to understand that a little bit to see if there's residual processing. But when you start looking at your other biocompatibility, like implant and hemocompatibility, that is the reason that the biggest impact is those surface characteristics. And so that's where those become very important. And using things like SEM, scanning electron microscopy, and some of the hemocompatibility tests, and an implantation test helps mitigate risks from surface characteristics.
Okay, great. Well, thank you, Thor, and thank you everyone for attending today. Unfortunately, we're out of time, and we like to thank Thor for sharing his expertise with us. And as a reminder, you'll be able to access this webinar and any of our previous webinars on demand under the Education tab on nelsonlabs.com. You'll be able to access the recording of this webinar in about a week. And as Thor mentioned as well, he also wrote a white paper on this topic, and you'll be able to find that under the Education tab under white papers as well. If you have any additional questions for Thor, you can contact him by email, or if you need any pricing information or a quote, please contact our sales department at sales@nelsonlabs.com. Again, we'd like to thank everyone for attending today. We hope you have a great day and stay safe.